Clear questions for Chapter 10, the nucleus. Number 1, lamins are members of a class of structural cytoskeletal proteins called intermediate filaments. Microtubules and microfilaments are other components of the proteinaceous cytoskeleton. What is the major difference between lamins on the one hand and microtubules and microfilaments on the other? A. Lamins are non-force generating and structural only. B. Lamins are found in both animal and plant cells. C. Lamins are located exclusively inside the nucleus. And D. Lamins associate with each other. The other proteins act as monomers. Answers A and C actually answer this question best with regards to the differences between lamins and microtubules and microfilaments. So as you can see, lamins, like intermediate filaments, are going to be mostly non-force generating and structural only, whereas microfilaments such as actin and myosin in some muscle cells can actually generate movement, and the microtubules will actually have movement along its own carrier proteins along the microtubule itself. Lamins are known not to do that. Lamins are found in animal and plant cells just like microtubules and microfilaments. That is a similarity. However, lamins are located exclusively inside the nucleus, which is true. Lamins do associate with each other. In fact, they form dimers and tetramers, and the other protein acts as monomers, but very similar to other microtubules and microfilaments. 2. Transport through a nuclear pore complex is the only way for molecules to enter or leave the nucleus. True or false? This answer is simply true. That's the entire purpose for the nuclear pore complex, is to control what molecules come in and which ones leave the nucleus. Both the large and small ribosomal subunits are assembled in the A, cytoplasm, B, nucleolus, C, nucleus, or D, endoplasmic reticulum. The answer here is B. So this is textbook. Large and small ribosomal subunits are going to be assembled in the nucleolus. Control over which proteins are imported into the nucleus relies on A, recognition of the protein by the nuclear pore complex, B, phosphorylation, dephosphorylation events, C, ubiquitination and degradation of inhibitory proteins, D, the hydrolysis of ATP, or E, all of the above. Of course, the answer for number 4 is E, all of the above. All 4 steps need to occur in order to import certain types of proteins, in this case transcription factors, through the nuclear pore. So in this case, we are looking to phosphorylize an inhibitory protein to allow the inhibitory protein to release and allow the transcription factor to be transported or first recognized by a nuclear pore. And this also involves other phosphorylation and dephosphorylation events, as well as the ubiquitination and degradation of the inhibitory proteins. Lamins bind to A, other proteins, B, lipids, C, chromatin, or D, all of the above. And the answer to number 5 is also all of the above, letter D. Lamins do bind to other proteins, lipids, and of course to different forms of chromatin, both euchromatin and heterochromatin. In order for the Fish technique to localize specific chromosome or regions, the chromosome must be A, separated, B, denatured, C, hybridized, or D, replicated. And the answer to number 6 is C, hybridized. If you know what FISH stands for, which stands for fluorescence in situ hybridization, you know what kind of technique is used to localize these specific chromosome regions. So in this case, the chromosomes must be hybridized in order to be visible. If a FISH probe localized a particular exon, what could you conclude about the chromosomal region if it lit up in the assay? A, the chromosome region is made of euchromatin. B, the chromosome region is made of heterochromatin. C, the chromosome region should localize to the lamina. Or D, the chromosome region is being actively transcribed. The best answer for number 7 is D, the chromosome region is being actively transcribed. I feel like this question needs to be specialized a little bit more closely here. So I think if you saw where the actual chromosome region is lit up in the assay, if the chromosome region was closer to the center or the most internal portion of the nucleus, I would say yes, it is being most actively transcribed. However, it's found more towards the periphery. It tends to be more of an inactive domain, so this could also be least actively transcribed. Again, depending if you know what gene that you're looking for, generally if it's closer to the center, usually will be more actively transcribed and technically be more euchromatin, but then if you found it in the periphery very close to the edge of the nuclear envelope, then you could argue that it could be a region made of heterochromatin and very unlikely to be transcribed. Number 8, replication factories are distinct in that they contain A, areas of replication and multiple DNA molecules, B, multiple copies of RNA polymerase 1 and 3, C, high concentrations of lemon-binding proteins, or D, transcriptionally inactive chromatin? The answer is A. Of course, if you found a replication factor, you assume it would be replicating DNA, so A is your best answer. Areas of replication of multiple DNA molecules. You would not normally see RNA polymerases 1 and 3. Of course, high concentration of lemon-binding proteins usually means it's going to be heterochromatin, so least likely to be transcribed. And of course, letter D will follow with that. So transcription inactive chromatin would not be an area where we would find a replication factory. 9. The discovery of chromosome territories required the development of powerful microscopes. True or false? Number 9, the answer is false. We do not need powerful microscopes. We just need different types of techniques, like FISH, defluorescence, and pseudohybridization. Very simply, we can find or label the oligonucleotide probes with complementary to specific repeat sequences. You can very easily visualize which territory different types of chromosomes we expect to see within the cell. And again, we can also determine whether or not if they are in the euchromatic regions or the heterochromatic regions. Number 10, chromosomes occupy discrete regions of the nucleus. This is called A, chromosome factories, B, chromosome localization factors, C, chromosome lemon-binding proteins, or D, chromosome territories. Answer number 10 is D, chromosome territories. This is actually a textbook for what the chromosomes and regions they occupy are simply termed as the chromosome territories. Number 11, what would happen if a nuclear body were treated with a protease? A, it would be exported from the nucleus. B, its chromatin would condense. C, its membrane would dissolve. Or D, is likely to fall apart. The answer is D, it is likely to fall apart. So very simply, a nuclear body, if it's composed mostly of proteins, of course a protease is going to be a proteolytic enzyme. It will break down individual proteins, and therefore a nuclear body will break itself apart. 12. Because they are not bound by a membrane, nuclear bodies can freely exchange proteins and RNA molecules with the rest of the nucleus. True or false? Number 12, the answer is true. Very simply because there is no membrane around the nuclear body, it can freely exchange proteins and RNA molecules with the rest of the nucleus. Number 13, the nuclear organizing region is an association between A, the nucleolus and the nucleus, B, the nucleolus and the euchromatin, C, the nucleolus and the heterochromatin, or D, the heterochromatin and euchromatin. The best answer for 13 is B, nucleolus and euchromatin. So very simply, the definition for the nuclear organized regions are going to find the genes for the different sizes of the ribosome RNAs, in this case the 5.8S, 18S, and the 28S. So therefore, these are going to be highly transcribed regions, which include the euchromatin, as well as the nucleolus. The other answers, nucleus and nucleus, wouldn't make any sense. Nucleus and heterochromatin is going to be very inactively transcribed, and of course heterochromatin not being actively transcribed would not fit letter D as well. Number 14, the difference between a polycomb body and a transcription factory is that A, transcription factories are associated with euchromatin, polycomb bodies are associated with heterochromatin. B, polycomb bodies are sites of transcriptional activation, transcription factories are sites of active repression. C, polycomb bodies are sites of transcriptional repression, transcriptional factories are sites of active transcription. Or D, transcription factories are associated with heterochromatin, polycomb bodies are associated with euchromatin. Now the answers for 14 are very similar. In this case, A and C would qualify as answers for this question. If you read through both, they'll have very similar definitions here. So the transcription factories are associated with euchromatin, which you know are going to be actively transcribed. And of course, the polycomb bodies are going to be associated with heterochromatin, which you know is going to be somewhat inactive. So in letter C, it pretty much talks about the same thing, just in reverse order. Polycomb bodies are sites of transcription repression, so you know they're not actively transcribed, whereas the factories are sites of active transcription. And very simply, the answers B and D would be the opposite of what you see for A and C, so of course they will not be applicable answers. 15. What do the various nuclear bodies have in common with cytoplasmic organelles? A, all are bound by membranes that help them isolate and regulate specific metabolic pathway reactions. B, all represent localized and isolated areas where specific metabolic reactions can take place and be controlled. C, they can freely exchange substrates and products with the surrounding medium in order to direct and speed up specific pathways. Or D, they each have mechanism for membrane-based transport, regulatory steps that control what gets into and out of the organelle. Letter B, all represent localized and isolated areas where specific metabolic reactions can take place and be controlled is our best answer. Letter A, the statement all are bound by membranes that help isolate and regulate specific metabolic reactions. Very simply, that's one of the definitions for various nucleotides is they have no membranes to contain those particular areas. The various nucleotides do exchange substrate and products within surrounding medium. However, the cytoplasmic organelles do not, especially ones that are born with the endomembrane system. They release things through vesicles from one to the next, in this case the ER, to the Golgi apparatus, and then possibly to a different part of the cell, as well as to different areas of the plasma membrane. Letter D is false, since they only the membrane-bound organelles of the cytoplasm have membrane-based transport, whereas the various nucleotides do not.